Okay, so we're going to just continue our discussion of proteins by talking about the secondary structure of a protein. Remember, the primary structure is just the amino acid sequence in order from N terminus to C terminus. So what is the identity of each of those amino acids in the chain? But proteins aren't just long chains. Everything about a protein corresponds to its fold. Protein folding is a big area of research. So these chains kind of like fold back on themselves, kind of like a crinkled up bundle of yarn. And that fold is what dictates the overall action of that protein, its function. So secondary structure are going to be these common structural elements that we see in a lot of proteins. Over and over and over again, we see these common structural features. And then we'll talk about tertiary structure next after this, and that's kind of the overall shape of the protein. So these secondary structural features, they're kind of like the beams and trusses that go into making a roof, right? All roofs have beams and trusses, but not all roofs are the same size. So they're these kind of like building block elements that we see over and over again. And there's two main types that we're going to focus on. There's three. There's two main ones though: the alpha helix and the beta pleated sheet, or simply referred to as a beta sheet. Okay, what we're going to see is these secondary structural elements are so common because they're held together by common hydrogen bonding patterns within, or rather between amino acids. Okay, and then these are our secondary structural elements. Oops, that's going to be too big. And then they will contribute to the overall tertiary, and again, we're going to do a whole lecture on this, but tertiary structure of the protein. So given the tertiary structure of a protein, we can identify here's an alpha helix and here's a beta pleated sheet. So again, these are just kind of like common structural features that we're going to see over and over again in our proteins. All right, so the first one is an alpha helix. So an alpha helix is something that's very common. The identity of the amino acids that make up the alpha helix aren't really all that relevant. So any combination of amino acids can make up an alpha helix, with the exception of proline, because remember, that's got that weird kink going on. And why is that? Because it has everything to do with that amino acid backbone, right? So all amino acids have this commonly repeating feature. Here's my alpha carbon, and then now I have my other carbonyl carbon, et cetera, alpha carbon, right? And so this is my R group. So all amino acids have this same kind of chunk to them when they're in a polypeptide chain. They have a carbonyl carbon, and then they also have a nitrogen that has a hydrogen bound to it that is capable of hydrogen bonding. And so what happens is these amino acids in our polypeptide chain will sort of curl up and form a helix, and they'll do so in such a way that the carbonyl carbon of one of our amino acids is forming a hydrogen bond with the nitrogen, that nitrogen hydrogen of the amino acid on the next rung down of our helix, right? So then the neighboring carbonyl carbon has that same hydrogen bond with that nitrogen oxygen, et cetera, all the way down. And so you can see how this is going to be this common, it has everything to do with just the backbone of a peptide, of a polypeptide, which contains that carbonyl carbon and the nitrogen single bonded to a hydrogen. These are able to hydrogen bond to one another. That's what this dotted line represents here, are these hydrogen bonding takes place. And that holds this peptide chain in a helical structure. And then the R groups would be kind of like sticking off to the side. So here the R groups are in purple. And again, the identity of the R group isn't really important, but they're going to be kind of pointed out away from the helix going down. All right, so the backbone, which is, if I'm looking at my alpha helix, what do I mean by backbone? Let me just clarify. The backbone is the carbon, nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, et cetera, of this polypeptide chain. That's twisted around, and you have the carbonyl carbon of, or the carbonyl group of one amino acid hydrogen bonding with the nitrogen hydrogen of the other amino acid, of another amino acid. They tend to be 3-4 amino acids away. A similar hydrogen bonding pattern is seen in beta pleated sheets. So again, this has everything to do with just the interaction, hydrogen bonding between the peptide backbone. So here is my polypeptide. Again, the R groups are represented by those purple spheres. And we can see that we have the carbonyl group of one amino acid hydrogen bonding with that hydrogen nitrogen of the other one, right? So that makes up, that's in that peptide bond. Likewise, then we move one down and now it's reversed. We got that hydrogen nitrogen, hydrogen bonding with the carbonyl carbon, et cetera, going down the line. Okay. And what you see is now the R groups are sort of sticking out of the plane of this sheet, right? So these forms what are called beta pleated sheets. Kind of like, you could see this sort of like almost zigzaggy pattern that's formed between the backbones. And then the R groups are sticking either up or down, right? So above the plane, sticking out at you, these R groups. Whoops. These R groups are pointed out at you, sticking out of the page, and then every other R group is now pointed into the page as well. So these ones back here are pointed into the page. Okay, so these secondary structures form these kind of flat sheets where the R groups are either pointed up or pointed down, depending on which amino acid. So every other one is pointed up, and then the ones in between are pointed down. Okay, so these are these two common structural features that you see in all sorts of different proteins, whether they're transport proteins or, you know, I don't know, all the other different classes, right? So the enzyme, any enzyme, whether it's in the liver or the heart or wherever it may be, they all tend to have these same structural features, the alpha helices and the beta pleated sheets. Okay, so just an example. This is a protein that I actually studied when I was in college, and so you can see there, I don't know, this kind of big intimidating structure you got here. But if you zoom in, we can see these same structural features kind of repeating themselves over and over, right? We see the alpha helices, and then there are even a few beta sheets. This particular protein has a lot more alpha helices than beta sheets, but we can see a few, for example, my pointer's not gonna work here. But those are those arrows. This is what would be called like a ribbon model of a protein. Okay, and that ribbon model is used specifically to show these different secondary structural features, right? So again, this is the sort of example that we started out with. We have these alpha helices, and we know now that these are what held together by hydrogen bonding from the peptide backbone, and we can spot them in the larger structure of our protein as well as these beta pleated sheets. And that's what makes up kind of the core of this particular protein that's pictured here. You also have these other regions, right? And these are just unstructured regions. They don't fall into one of those secondary structure categories nice and neatly. And those exist as well, right? The point of the secondary structure is they're just these structural features that we tend to see over and over again when we study protein structures. The last one is one that's fairly rare because it's a feature that's predominantly seen in structural proteins. So things like collagen and the tendons and, the outside layers of your skin and stuff like that. So these are structural proteins, not enzymes that perform like chemical functions or transport functions or anything like that. These are the ones that kind of just stand there and hold things into place. And they have these secondary structural features called a triple helix. So that's where you have 3 alpha helices that are kind of like braided together or wrapped out around one another to form a triple helix. And again, these are going to be features of structural proteins. All right, so let's see if we can't categorize, kind of match these here based on what we learned. We're going to try to match, it's going to either be the primary structure, an alpha helix, a beta pleated sheet, or a triple helix. The polypeptide chains that are held side by side with H bonds being hydrogen bonds. So which one would those be? That is the characteristic of a beta sheet. The sequence of amino acids in a polypeptide chain. So that's not a secondary structure, that would be the primary structure of the protein. A corkscrew shape with H bonds between amino acids. That corkscrew structure is referring to an alpha helix. And then 3 polypeptide chains woven together like a rope. That's a feature of the triple helix. Whoops.